And we are live.
Good morning, everyone, wherever you are working from the office or remotely, as many of us are today. I want to thank you for joining us for this webinar on cell therapy treatments for COVID-19. My name is Michael Lemke. I'm the Director of Science and Industry Affairs at the Alliance for Regenerative Medicine. The Alliance for Regenerative Medicine, or ARM for short, is an international advocacy organization. We are dedicated to realizing the promise of safe and effective regenerative medicines for patients around the world. We currently have over 350 member organizations, 2 of whom will be speaking today, small and large companies, not-for-profit research institutions, patient organizations, and other sector stakeholders across 25 countries. Our priorities include advocating for clear and predictable regulatory pathways, enabling market access, addressing industrialization and manufacturing hurdles, and compiling sector data in order to educate the media and other stakeholders. I'd like to call your attention to some resources on our website at www.alliancerm.org. These include future webinars in our Cell and Gene webinar series, our COVID-19 resource library, which provides up-to-date resources for stakeholders on who are managing R&D, operations, finance, and other aspects of their mission during the COVID-19 pandemic, our CEO discussion forum, and our weekly newsletter, which now includes items that are specific to COVID-19 leading the newsletter. During the webinar today, I'll make a brief introduction. We'll have 2 speakers presenting on the use of cell therapy treatments for COVID-19. There will be an audience Q&A session at the end of the webinar. You can submit questions anytime using the Q&A tab on Zoom. Please do use that Q&A tab and not the chat, as I will not be checking chat regularly during this webinar. Some brief background. Currently, academic research centers and therapeutic developers, some of whom are shown here, announced that they're investigating the application of cell therapies to treat COVID-19 and related complications. We are currently tracking over 24 clinical trials in this area and an additional 25 programs which are in preclinical development. Many of these developers are utilizing MSCs and other stromal cells to treat acute respiratory distress syndrome, which is a major cause of mortality in COVID-19. MSCs have been shown in other indications to manage severe inflammatory responses and cytokine storm, similar to that seen in patients with ARDS caused by COVID-19. Our speakers today are Dr. Eric Jenkins, Senior Medical Director and Head of Clinical Operations at Athersys, and Dr. Raquel Ophir, Vice President, Research and Development and Intellectual Property at Pluristem Therapeutics. I'll introduce them in a moment, but first I wanted to give you a brief plug for our next webinar in our Cell and Gene webinar series, which is on the topic of cell and gene therapy market access in the COVID-19 world. That will take place next Wednesday at 12 p.m. Eastern Time. You can find more information and registration information on our website. Finally, for those of you that want to dig more deeply into this topic, I want to call your attention to ISCT's virtual meeting next week, which will include 2 extensive sessions on COVID-19, including presentations by healthcare workers on the front line and their experience, and also presentations on new and novel therapies for COVID-19. I would encourage you to check that out. And with that, I will introduce our first speaker today, Dr. Eric Jenkins from Athersys. He's the Director and Head of Clinical Operations there, where he's responsible for medical oversight and all clinical development activities. And he'll talk to us today about the multi-stem product and its use in COVID-19 patients. Dr. Jenkins?
Thank you. I'm going to try to share my screen here. All right, thank you. First of all, on behalf of my colleagues and the entire Athersys team, I'd really like to thank everyone for taking part in the meeting today and to thank ARM for inviting me. I'm going to tell you a little bit about our ARDS development program that we've been working on for several years now, and for which our current COVID-19 induced ARDS pivotal trial is the most recent culmination. As part of this presentation, I will be making some forward-looking statements that are covered under Safe Harbor. These are the key topics that I'll cover today in the presentation. In order to stay within my allotted time and allow for discussion at the end, I will move through some segments of the presentation pretty quickly. I do have a fair amount to cover as we've been developing the use of multi-stem cell therapy for acute respiratory distress syndrome for quite some time. This spans the gamut from cell characterization and development of deep understanding of the cell's interaction with the complex processes and cellular elements of the host immune system during inflammatory stimulation, through preclinical testing in ARDS disease models, and promising clinical trials in moderate to severe ARDS patients. And now, most recently, our discussions with BARDA leading to expedited planning, FDA authorization, and rollout of a pivotal phase 2/3 trial to investigate multi-stem treatment of ARDS due to COVID-19. COVID patients who succumb to this disease, in the vast majority, are dying from complications of acute pulmonary inflammation that the virus causes. This is the same phenomenon that has been previously observed with other emergent pathogens. Currently, there is no effective treatment for acute respiratory distress syndrome beyond supportive care. While intensivists have made continual refinements in the standards for provision of expert, meticulous care of these patients, we have nothing to offer them in terms of specific treatment for the hyperinflammatory pathophysiology of ARDS. Preclinical and clinical evidence that I will present today shows that IV administration of multistem, our innovative off-the-shelf cell therapy treatment, can mitigate the hyperinflammatory cascade causing ARDS and help restore pulmonary function for severely ill patients. Importantly, multistem is not pathogen specific, which means it has broad potential relevance for situations where acute severe pulmonary inflammation occurs. From a regulatory standpoint, multistem has received RMAT designation, which is akin to the more familiar breakthrough therapy designation, but specifically for cell therapy and regenerative medicine treatments. As well, Athersys' multi-stem cell therapy has received 3 fast-track designations for specific indications from the FDA, including most recently for ARDS, based on very encouraging phase 2 clinical trial results demonstrating multi-stem's potential to fulfill a significant unmet medical need for treatment of this life-threatening condition. While our most advanced program is currently in ongoing phase 3 studies for treating ischemic stroke patients here in the US and internationally, we have been working for several years now in the ARDS indication with leading translational and clinical acute pulmonary and critical care researchers in the US and UK. We believe strongly that the responsible way to develop cell therapies is through close collaboration with regulators such as FDA and its international correlates, through programs, designations such as RMAT, to develop scientifically robust, well-designed, adequately controlled and blinded, randomized clinical studies that permit valid assessments of safety and potential effectiveness of these therapies for patients. This approach holds even in settings such as the current outbreak, where rapidity of spread and lack of known effective treatments can lead to desperation and urgency in the face of unmet therapeutic need. A few key points on the technology itself. First, the special class of cells that comprise multistem have very robust growth properties and a distinctive immunological profile. They are not MSCs. They have different cell surface markers, gene expression profiles, developmental differentiation capacity, and immunomodulatory phenotype, and are smaller cells with greater ex vivo proliferative capacity. We obtain our starting material for the product from bone marrow of healthy consenting adult volunteers. Because of the unique ex vivo proliferative properties of these cells, we can subsequently produce millions of clinical doses with material obtained from a single healthy donor. The product has very long shelf life. We now have over 8 years of stability data on our cryopreserved product. Preparation and administration of the product is very straightforward. The vialized product is thawed over just a few minutes, transferred to a bag of saline diluent, and infused intravenously over about 30 minutes to an hour. In typical years, acute respiratory distress syndrome, or ARDS, affects 500,000 patients per year in Europe, the United States, and Japan combined. Though as we now clearly know, an outbreak of highly virulent respiratory pathogen can result in a surge in ARDS incidence. With no currently available pharmaceutical or biological agents to effectively treat the hyperinflammatory lung injury, ARDS represents a major area of unmet medical need, one with high mortality and morbidity, typically requiring extended, resource-intensive ICU stays, and among survivors, an arduous recovery during convalescence, often leaving them with long-term disabilities and other sequelae. In most cases of viral pneumonitis, patients experience an initially exuberant inflammatory response that, coinciding with viral clearance, transitions into a convalescent or resolution phase, which supports healing of injured tissues and clinical recovery from respiratory illness. However, in a subset of individuals, this restoration of homeostasis does not occur. Instead, hyperinflammatory immune responses persist, resulting in ARDS pathology and necessitating prolonged mechanical ventilation of the patient. Based on our clinical results and other data, we believe that in these vulnerable patients, multi-stem can effectively interrupt this pathological cycle and promote an immunologic transition that leads to patient recovery. Multi-stem, which is composed of living human cells, interacts dynamically via both paracrine and direct cell-cell interactions with the host immune system and injured tissues. It does so through multiple well-defined mechanisms of action that depend on the tissue environment into which they're introduced. Prior to and in parallel with our ongoing clinical development programs for multi-stem, researchers at Athersys and in collaboration with multiple external research groups have rigorously investigated the mechanisms by which multi-stem's immunomodulatory properties interact with the host's immune system. Multi-stem alters host immune cell differentiation, proliferation, activation, and function, helping to tip the balance from a hyperinflammatory immune effector cell-dominated response toward more tolerogenic, anti-inflammatory, and reparative immune system functions. These are the processes that normally predominate during the later convalescent phases of the immune response to tissue injury. In this way, multi-stem facilitates restoration of immune homeostasis during illnesses in which a deleterious, persistent hyperinflammatory state could otherwise perpetuate a cycle of secondary tissue injury. ARDS is well known as just such an illness. A combination of infectious, chemical, or mechanical lung injury is exacerbated and then really overtaken by, as depicted in this figure, the many redundant pathways that lead to hyperinflammatory lung self-injury, resulting in the diffuse alveolar damage that is pathognomonic for ARDS, the accumulation of proteinaceous alveolar edema, diffuse interstitial inflammatory infiltrates, hyaline membrane accumulation, and early and late interstitial fibrosis. Specific mechanisms by which multi-stem is understood to interrupt this hyperinflammatory response include reduction of neutrophil tissue infiltration via downregulation of cell surface adhesion molecules such as E-selectin, VCAM, and ICAM on vascular endothelial cells, as well as by moderating a shift in neutrophil populations from a pro-inflammatory phenotype towards a predominantly anti-inflammatory phenotype, resulting in decreased neutrophil activation, apoptosis, degranulation, myeloperoxidase secretion, and netosis. Multi-stem also results in a shift in the monocyte macrophage lineage from the M1 towards an M2 phenotype, resulting in enhanced macrophage phagocytic activity, decreased accumulation of inflammatory cytokines such as IL-1 beta, IL-6, and IL-8, and upregulation of anti-inflammatory cytokines, including IL-10 and TGF-beta. Similarly, we observe a decrease in CD3 positive T effector cells and increased numbers of T regulatory cells, thus mitigating T cell dependent potentiation of the hyperinflammatory microenvironment within the alveolus. By affecting these shifts in the immune response, multi-stem can break the deleterious cycle of self-perpetuating immune-mediated tissue injury and promote a more rapid restoration of immune homeostasis, ultimately allowing clearance of alveolar edema, both through reduction of alveolar capillary endothelial barrier permeability, as well as restoration of active energy-dependent alveolar fluid clearance by AT1 epithelial cells. To evaluate the potential for human clinical trials of multi-stem for treatment of ARDS, we undertook preclinical studies to investigate the organ-level effects in disease models of inflammatory lung injury. Large animal LPS-induced lung injury models, not shown on this slide, demonstrated multi-stem treatment restored normal oxygenation following injury. We also, as this slide depicts, evaluated the effects of multi-stem administration to ischemia reperfusion injured human donor lungs that had been rejected for transplant due to the resulting inflammation and corresponding impairment in gas exchange. In this model, following 8 hours of cold ischemia, donor human lungs were placed onto warm perfusion ventilation systems. The contralateral lung of each lung served as its own control. So multi-stem was administered into the left lower lobe, while the right lower lobe received saline. Blinded histopathological inflammation scores are depicted for each lung pair on the left figure of this slide. As you can see, there's a consistent reduction in inflammation scores in the multi-stem treated lung compared to the contralateral saline control. This can be visualized in the representative photomicrograph on the right. On the basis of these and other studies, we advanced multi-stem into clinical development for the treatment of ARDS. The MUST-ARDS trial was an exploratory phase 2 study conducted at leading pulmonary critical care hospitals here in the United States and in the UK. Because this was the first time multi-stem was administered intravenously to very vulnerable human subjects with critical underlying hypoxemic respiratory failure, and frequently with other comorbid organ failures and hemodynamic instability requiring vasopressor support, we took a cautious approach. The study incorporated 2 small open-label dose escalation safety cohorts. In cohort 1, 3 subjects each received a single infusion of 300 million cells within 96 hours of ARDS onset. This dose appeared to be well tolerated, and DSMB safety review recommended proceeding to cohort 2, in which subjects received 900 million cells. When this dose evaluation was also well tolerated, we proceeded to cohort 3, in which 30 subjects with moderate to severe ARDS, i.e. a PaO2-FiO2 ratio less than 200 millimeters of mercury, were randomized in a double-blind manner to receive multi-stem treatment or placebo in a 2-to-1 ratio. The primary outcomes were evaluated at the traditional 28-day time point, which is a standard time point for ARDS clinical trials. But the study continued for a full 1 year of follow-up to evaluate both long-term safety as well as patient-centered quality of life outcomes. 28-day outcomes are summarized on this slide. It's important to note that this study was an exploratory study and not powered for efficacy outcomes, with the primary endpoints being safety and tolerability. So first and foremost, IV administration of 900 million multi-stem cells demonstrates consistent safety and tolerability in this critically ill patient population, with the incidence of treatment-emergent adverse events comparable between treatment groups. Importantly, there were no treatment-related serious adverse events in this trial. 28-day mortality outcomes substantially favored the multi-stem treatment group, as evidenced by the 40% mortality rate in the placebo arm, a rate typical for patients with moderate to severe ARDS, versus only 25% mortality in the multi-stem treated patients. Importantly, meaningful differences were observed in ventilator-free days and ICU-free days, also favoring the multi-stem treated patients. Among patients with more severe ARDS, which was a prospectively defined population and analysis for the trial, we observed even larger differences in mortality, ventilator-free days, and ICU-free days, again, all strongly favoring the multi-stem treatment group. As a whole, this and other data from the study suggests that multi-stem treatment appears to meaningfully improve pulmonary function, expedite weaning from invasive ventilation, enhance patient recovery, and improve survival of patients with ARDS, particularly among the more severely affected patients. Serum biomarker concentration results support our understanding of the immunomodulatory mechanisms of action of multi-stem, and were consistent with effects seen in our previous phase 2 clinical trial, the MASTERS-1 study, evaluating treatment of ischemic stroke treated with multi-stem. Our clinical development partner in Japan, Helios, is currently running a trial of multi-stem in ARDS patients. In their OneBridge trial, they are limiting enrollment to subjects with intrinsic lung injury, such as bacterial or viral pneumonia, who have developed ARDS with severe radiographic features by high-resolution CT. At their behest, we performed a post hoc subgroup analysis of ARDS study subjects with intrinsic pneumonia-induced ARDS and severe impairment in gas exchange. This subset, probably most relevant to the current situation with COVID-19 viral pneumonia-induced ARDS, again shows consistent and meaningful improvement in mortality, ventilator-free days, and ICU-free days following multi-stem treatment. While the primary safety and efficacy outcomes were evaluated at the 28-day time point traditionally used in critical care and ARDS trials, we looked for earlier evidence of potential effectiveness, as well as the long-term patient-centered outcome of quality of life among survivors. Consistent with the previously described mechanisms of action, multi-stem treatment was associated with rapid improvement in lung function, with substantially higher probability of achieving ventilator independence by day 7 relative to placebo subjects. Specifically, 45% of multi-stem treated patients had achieved ventilator independence by study day 7, compared with just 20% of placebo recipients. Sorry about that. After 1 year, we observed evidence that among survivors, multi-stem treatment was associated with better patient self-assessed quality of life. Specifically, 80% of multi-stem treated survivors reported complete independence in self-care by 1 year, compared with only 40% of placebo-treated survivors, the remainder of whom required at least some assistance with bathing and dressing. In summary, we and the clinical investigators were quite encouraged by the results of this trial. And based on the preclinical and this clinical data, the ARDS program garnered our 3rd Fast Track designation from the FDA. So it was within the context of having presented these data over the course of the past year, starting with top-line 28-day results at the American Thoracic Society Conference last May, followed more recently by the 1-year follow-up data and quality of life assessments late last year, that in January, very early in the outbreak, we were approached by leadership of the Biological Advanced Research and Development Authority, or BARDA. They were aware of our RMAT and ARDS Fast Track designations and wished to discuss our technology as a potential treatment for the most severe pulmonary disease manifestations during emergent outbreaks of highly pathogenic respiratory infections. Importantly, multi-stem, with its mechanism of action being modulation of the host immune response, is pathogen agnostic, and thus relevant to not only SARS-CoV-2, but to other infectious and even non-infectious causes of severe systemic and pulmonary inflammation within BARDA's purview. With a long shelf life, unique scalable manufacturing, simple IV administration, and demonstrable tolerability, even in the most critically ill patients with cardiopulmonary failure, they, I think, had the foresight to see that multi-stem could very well fulfill a significant need gap within our strategic preparedness for emerging disease threats. They seek, and we believe multi-stem could represent, an effective treatment for severe pulmonary inflammation, one that could be at the ready, even in the opening days of outbreaks like the one we're facing now, before specific anti-infective agents, or certainly vaccines, could be developed, tested, mass manufactured, and distributed. These initial discussions were followed by a request that we present our data and a proposal for a pivotal trial of multi-stem for treatment of COVID-19 induced ARDS during the first TechWatch-style meeting of the interagency US government CoronaWatch Medical Countermeasures Task Force. Our program proposal was graded as highly relevant, and we've been in continuous ongoing dialogue with the office since that time. We've not wasted any time, however, with discussions around potential BARDA contract support ongoing. We've been hard at work getting our trial underway, collaboratively interacting with the FDA on design of a robustly powered, randomized, double-blinded, placebo-controlled pivotal study of multi-stem for moderate to severe ARDS and COVID-19 patients. This is the same indication in which we already have encouraging, randomized, placebo-controlled phase 2 data supporting tolerability and potential effectiveness in this critically ill and especially vulnerable patient population. Our interactions with the FDA have been very productive, with extremely fast turnaround by their subject matter experts to provide helpful feedback on our proposal and our responses to their queries. We were able to incorporate their input into a trial design and obtain authorization to proceed with recruitment of a pivotal trial on what, in my experience, is really an unprecedented timeline. Hats off as well to our clinical investigator collaborators and to our CRO colleagues at MedPace, who provided amazing flexibility and an awful lot of really hard work leading up to the trial startup. We have begun enrolling patients into the MACOVIA study, which is a phase 2/3 trial designed to treat a patient population very similar to those from our MUST-ARDS phase 2 study with moderate to severe ARDS. We will administer 900 million to 1.2 billion multi-stem cells or placebo intravenously within 48 hours of developing ARDS with profound hypoxemic respiratory failure, defined by a PF ratio less than 200 millimeters of mercury. We have incorporated plans for patients with persistent ARDS without evidence of rapid improvement, being eligible to receive a second dose 3 days after the first dose. Because of the evolving standards of care in this emerging pandemic response, we've taken a pragmatic approach to accommodating novel COVID-19 therapies as concomitant medications. I think we've developed an approach that preserves trial data integrity, yet will allow clinicians the option to provide novel therapeutic strategies as evidence of their potential effectiveness emerges, and still offer participation in our trial if and when their patient's condition unfortunately deteriorates such that they meet eligibility. We have incorporated pre-planned interim analyses by independent data and safety review board who will make recommendations about adjustments to study design and potentially sample resizing. We are committed to working in consultation with our colleagues at the FDA when it comes time to evaluate potential trial actions or adjustments in response to the independent DSMB's recommendations. The primary endpoint agreed to by the FDA is a group comparison of subjects' ventilator-free days score, which is a composite endpoint of liberation from mechanical ventilation and mortality over 28 days post-randomization. Secondary endpoints will include 60-day all-cause mortality, ICU-free days, and the proportion of individuals achieving early ventilator independence by day 7. We will also assess an extensive array of biomarker responses, including cytokines and specific immune cell populations, as well as 1-year follow-up outcomes of patient self-assessed quality of life by a validated survey instrument. So I know I went through a lot of material really quickly. Most of these slides are available on our company website for those who want to review the material at a more leisurely pace. And I'll now hand it off to Dr. Ophir and look forward to her presentation and any further discussion to follow.
Thank you, Eric. That was a great presentation. So next up, we'll hear from Raquel Ophir. Again, she is the Vice President of Research and Intellectual Property at Pluristem, and she will tell you about their placental-derived product and its use to treat COVID-19.
Thank you very much, Mike and the ARM, for inviting me to give this presentation. Thank you very much, Eric, for this fascinating talk. It's always very pleasant to listen to people who are thinking very much alike and have similar approach. So it's very comforting to know that people have similar approaches, even though there are some differences. So I'm Raquel Ophir, and I'm from Pluristem, and I would like to share today our approach to treating COVID-19 patients. So this is a forward-looking statement, and I will move directly to the presentation. So just a few words about Pluristem. We're an Israeli-based cell therapy company focused on regenerative medicine. We have several off-the-shelf placenta-derived cell product candidates. We have 2 ongoing phase 3 studies in ischemia associated with diabetes complication and in muscle regeneration. We have favorable safety profile and efficacy data from hundreds of patients treated worldwide in these 2 indications. We have our own cell manufacturing technology, which we are very proud of, and we have spent a lot of efforts in developing it in the past few years to produce very high quality of cell products in a commercial scales. We have strong IP. We're a publicly traded company, both in Tel Aviv and NASDAQ, and we are about 150 employees as for today. And the source of our cells is the human placenta. This is a very rich source of cells. It does not involve any ethical complications. It is a very rich and diverse source of cells. The cells are very potent cells. They have immunomodulatory properties as well as pro-angiogenic properties. The donors of the placenta are by definition young donors, and we have, of course, full selection criteria for the placenta donor. Happily, this is an unlimited source of cells and easy to collect, and we can manufacture very large quantities of cells from each placenta using our bioreactors. So just in a nutshell, we take the human placenta from maternity units here in Israel. We produce the cells in our in-house, in our bioreactors. At the end of the process, we get the cells cryopreserved in liquid nitrogen. These are allogeneic cells off the shelf. They're ready to use, and they're injected. One of our differentiating factors is administrating the cells via IM administration, which we believe provides very large advantage, especially in the COVID-19 patients. The cells, even though injected into the muscle locally, they work by paracrine mechanism of action by secreting factors that give a systemic effect of a systemic effect and a long-term effect. This is a Pluristem pipeline. As I said, we have with PLX-PAD, the most advanced product that we're talking about. We are in phase 3 clinical trials, critical limb ischemia and muscle regeneration following hip fracture. We have also another product, PLX-R18, which is developed for hematological failure, acute radiation syndrome, and also other indications. And the PLX-PAD is now being used for the COVID-19 project, which I'm going to talk about today. So if we're looking at a schematic drawing of the mechanism of action of the cells, we see that the cells secrete many factors and they change their secretion profile in response to signals from the environment. They secrete angiogenic factors in response to ischemic conditions. They secrete muscle regenerating factors in response to injury. They reduce oxidative stress, and they also have very strong immunomodulatory properties. We have so far used the cells mainly to ischemic and muscle injury diseases, but nevertheless, these diseases also involve immunomodulatory components. And based on these immunomodulatory components, we decided to move into treating COVID-19 patients and looking at the possible effect of PLX-PAD cells in this population. So I think I will go very briefly over the introduction. I think Eric went over it and we've all heard it many times. Most of COVID-19 patients have very minor symptoms. A small proportion gets very complicated, develop ARDS, and has high mortality rates. And PLX cells are allogeneic, mesenchymal-like cells with immunomodulatory properties that may prevent or reverse the dangerous overactivation of the immune system, therefore maybe reduce the severity of COVID-19, pneumonia, and other lung complications, and improve the outcome of these patients. We already have very good safety profile of PLX-PAD cells from our other indications, already hundreds of patients treated, and because of the immunomodulatory and anti-inflammatory properties of the cells, we thought they could be useful for treating COVID-19 patients. So just a little bit about these properties, and I will not go deeply into the science here, but all of the next slides that I'm going to go briefly over demonstrate the effect of PLX-PAD cells on modulating hyperactivation of the immune system. So we have seen the effect of the cells in reducing PBMC, activated PBMC proliferation, and inducing upelevation of the anti-inflammatory IL-10 from activated PBMCs. When we look at activated PBMC cells, we can look at the secretion level of pro-inflammatory cytokines from these cells following a co-incubation with PLX cells, and we see a very drastic reduction in the pro-inflammatory cytokines, TNF-alpha, interferon gamma, IL-17, CD25, and many others. Also, as shown by others, we have seen also the effect of PLX-PAD in upelevating the M2 anti-inflammatory form of macrophages, and on the other hand, reducing the M1 population of activated macrophages. Also looking into the T cell population, elevation in the regulatory T cells that are the ones responsible in controlling the inflammatory response, and reduction in the pro-inflammatory Th1 population. And much of this data was very recently published with our colleagues in Italy, the group of Ornella Parolini, and it is available online. Also in works that we have done with the Charité, we have seen also reduction in the priming of natural killer cells and also in Th1 activation, all suggesting immunomodulatory effect, which we have also seen in our preclinical and clinical studies for other indications. Therefore, the suggested mechanism of action that we're proposing for PLX in COVID-19, the virus, as we know, attacks, and this is very schematic, but still, and through ACE2 receptors, which by the way is not expressed by PLX cells, infects the epithelial cells of the lungs. Of course, the body fights and activates the immune system to fight the viral infection. And then there are two outcomes. In most of the patients, as we know, there's a resolution of the infection and with no other complications. But in the subpopulation that we have discussed, there is what we call the cytokine storm, overactivation of the immune system, which gets out of control and needs to be resolved. Therefore, due to the immunomodulatory properties of PLX cells, we believe that this cytokine storm can be stopped, and we have a lot of data to support this motion. And indeed, in an animal model of ARDS, we see very nice recovery of oxygenation level and reduction in lung fibrosis. This is a pleomycin-induced ARDS model done with Professor Tebau from Alberta University. So what is the status of PLX-PAD for COVID-19 project? Since we are in phase 3 clinical trials, we have full tox, pharmacokinetics, and pharmacodynamic studies completed. We have safety data for several doses from hundreds of patients with the same product, with the same mode of administration, the intramuscular administration, because we're already in phase 3 studies. We have data from the patients that have been treated under the expanded access per patient program in the US and Israel. And we have clearance of the FDA for our IND application for a phase 2 COVID-19 study. And looking at the data from the 8 patients that we have treated suffering from COVID-19, and this was recently published on May 14th. The patients have been followed for at least 28 follow-up, 8 patients, one of them in the US and 7 in Israel. And out of 18 patients that were treated so far under the compassionate use program. And what we have seen is 7 out of the 8 were alive after 28 days. This is 87.5 survival rate, all under invasive mechanical ventilation at the time of treatment. 75% no longer needed of any mechanical ventilation at the time after 28 days, and over 62% of the patients were discharged, of course, alive from the hospital. And as I said, we have clearance of our IND for the phase 2 clinical trial, and we are hoping to get this clearance in Europe very shortly as well. A few words on the design of the study. This is a phase 2 randomized double-blind placebo-controlled study. The population is hospitalized mechanically ventilated adult patients suffering from respiratory failure and ARDS due to COVID-19. It will be done in US and also filed in Europe. Altogether, it will have 5 arms, 28 patients per arm, altogether 140 patients. Doses will be single or double 300 million cells, and also 600 million cells given at a single administration, and the placebo groups respectively. As I said, we are administrating the cells via IM administration, and we have seen in many clinical and preclinical settings that IM administration provides a systemic effect. And we think that in this population of patients, IM injection provides a huge advantage due to the coagulation complications that are known to be in these patients. And at least in our point of view, IV administration in this specific population may be of risk. The primary efficacy endpoint is the number of ventilator-free days within 28 days. And we also have secondary and many exploratory endpoints that I will not go into them at this time. In terms of our manufacturing technology, as I said, we have an in-house manufacturing facility based on technology that was developed in-house in bioreactors. The system is fully controlled, fully closed, fully automated, and scalable. And we have marketing-ready, cost-effective, industrialized platform. One very important aspect of our technology is full control over the condition within the bioreactor, which allows us a batch-to-batch consistency between the different products. We also have taken care of the entire cold chain supply. So not only that we manufacture the cells in-house, we also take full care of the quality, cold chain storage, shipment, even a thawing device that was developed by us. So we have a full solution from the harvest of the placenta until the bedside of the patients. This is our cold chain. We have shipments already in place to worldwide. And because we have ongoing clinical trials, we already have cells available in different hubs in the US. So to summarize, PLX-PAD is readily available placenta-derived cell therapy. We have high safety profile from hundreds of patients treated worldwide. We have the ability to support damage repair of various systems in the body by regenerative and anti-inflammatory, I see a mistake here, capabilities. We have a strong scientific background, which we believe will support the activity of the cells, positive data from clinical trials in other indications in response to inflammation, ischemia, muscle injuries, radiation, and hematological failures. And we have promising data from the expanded access per patient program. And the cells are available already in the US in logistic hubs within the US. We have approval for clinical trials for other indications by various regulators, and we have a lot of governmental support from the US, Europe, and Israel for different clinical and preclinical programs. And this is the Pluristem team. I want to also mention two very close collaborators of us, the Charité team in Germany, Hans-Dieter Volk, Petra Raneke, Tobias Winkler, and our dear collaborators in Italy, the group of Ornella Parolini and her team. Both groups we collaborate with and have provided a lot of support for our development. Thank you.
Many questions. Unfortunately, I think more than we're going to have time for, but some very good questions. So we'll do our best to get to as many of those as possible. Several of you asked about, unfortunately, there were a few audio issues. We're still learning in these new times how to deal with Zoom. So I thank you for your patience, but I wanted to let everyone know that there will be a recording, including the slides of this webinar made available on ARM's website. So you can check that out later if you were not able to view or hear the entire presentation. So I'll start out with a question for both of you that came in actually before the webinar started. I thought it was a pretty good question about geography in the trials. And do you think that in different areas, local patient population factors or differences in standards of care could affect the outcome? And is that, if so, is that something that you were able to account for in the design of your trials for COVID-19? And maybe Eric, if I could direct that to you first.
Yeah, sure. You know, I think there are different standards of care across some geographic settings. Particularly what we've found is that there's some differences between US and UK, EU care, and that used in Japan. But the differences are subtle, I think. You know, overall, I think there's good acceptance of sort of the tenets of good ICU care for these individuals with moderate to severe ARDS, specifically lung protective, low tidal volume ventilation, proning, and in the setting of COVID-19, you know, exploratory use of some of these novel therapeutic agents. There are some subtle differences across geographies, but also throughout, you know, inter-site differences, even within the US, on use of things like non-invasive mechanical ventilation to assist patients with severe hypoxemia and avoid intubation. But I think overall, you know, the care is fairly standardized. And, you know, we found in our MUST-ARDS trial between the US and UK that folks were really following very similar standards of care. So I don't think that's a major impediment to working across geographies, at least between the US, UK, and EU. But I'm interested in Dr. Ophir's perspective on that as well, since they plan to be working both in the US and EU, I understand.
Yeah, so of course, as you said, this is a challenge in every clinical trial to get as much standardization between the different sites. And as you said, even here in Haifa, we have some differences between different sites. But nevertheless, as you said, the main issues are similar. And I know you cannot draw too many conclusions from the compassionate care program that we have done, but there even though there was very high variability in the patients themselves and in the standard of care, still some of the factors that we followed were very similar. So, and also due to the hopefully quick recruitment of the patients, I hope we won't need to use too many different sites, and maybe we will need to do some standardization of the standard of care, but I don't think it's a major issue.
Okay, thank you for answering those questions. We had quite a few questions about dose and differences in dose, and without speaking to any specific products, which I think you're probably not able to do, you know that there are quite a few people working in this area, different cell types, which are sometimes loosely grouped together as MSCs. Can you comment at all on how you arrived at the dose, and can you hypothesize on how differences in cell types might affect dosing?
Yeah, I think with the caveats that we're working with different cell types, the dose has been individualized, based largely on the preclinical work that's been done with, at each company. So, we've done extensive, in vitro and preclinical animal model work to arrive at the optimal doses. We've had essentially the luxury of working with a product that seems to be very safe, even at fairly high doses by IV administration, even in these critically ill patients. As we're administering these cells IV, they are going directly to the lung. That's the first vascular bed that those cells are going to encounter. And so, yes, going into the MUST-ARDS trial, that, was a potential concern. And that's why we took a very cautious approach. We didn't take safety for granted in that population in particular. But what we found was that the cells were very, very well tolerated at that 900 million cell dose. That dose was chosen based on the preclinical work that was done to really be well up on the plateau of the dose response curve in regards to LPS-induced large animal lung injury models. We've worked with a slightly higher dose of 1.2 billion cells in our stroke program, as well as in our upcoming trauma program. So a not too different dose, but again, that was based on animal models with our specific cell type. So I think it's really depends on the cell type you're working with and your experience with its immunomodulatory effects in specific disease indications.
Mm-hmm. Yeah, I agree with Eric that you have to learn from previous studies. I must say that the ability to extrapolate from animal studies to human studies is challenging. I would like to say that we can directly extrapolate from a mouse or even larger animals, but I think most of the experience comes from the clinical studies. And we do see reaching a plateau at some point with the doses. We try to differentiate between chronic and acute situations. This is why we're checking two doses here, the 300 and the 600, because of the severity and the acute nature of the disease. And also, as Athersys are also trying to see, checking the effect of repeated administrations. Maybe we need a longer lasting effect. So I think we are all in the same magnitude of doses, and each company has to find the fine tuning for its particular product for the specific indication.
Okay, thank you. We also received questions about manufacturing, and maybe Eric, in the interest of time, I'll just direct this one to you. It's a topic that's near and dear to my heart. But with respect to your ability to scale up, if there is a second wave and we have to consider that possibility, do you feel comfortable and let's assume the therapy does work. Do you feel comfortable in your ability to handle the patient load?
Um, yes, we're working towards commercial scale-up manufacturing using a stirred tank bioreactor system that would allow us to scale up quite dramatically to really large capacity manufacturing to address the patient population that presents with moderate to severe ARDS. Certainly, there's the potential with cryopreservation and a long shelf life to actually stockpile product in a strategic stockpile for availability to roll out quite quickly with a new emergent outbreak. So I think between those two attributes of the product, I think we certainly could address even really dramatic pandemic presentations of high pathogen respiratory illnesses in the future.
Okay, good. It's good to hear. Raquel, maybe a question for you. Several people submitted questions about a little bit of the information, the compassionate use information you disclosed, and I think it was 8 out of 18 patients. Are you able to say any more about the other 10 patients, or have you has Pluristem made a conscious decision to focus on the data from the phase 2 clinical trial and those outcomes going forward due to the anecdotal nature of the compassionate use?
Yeah, so I cannot provide any more information. Of course, we're a public company and we have certain circumstances where we provide you information. We are focusing on the clinical trial now, and this is the main focus now.
Okay, yeah, I appreciate that, and I think you're doing the right thing there. I just thought it was appropriate to address that question.
No, it's a fair question. I
Okay.
understand curiosity.
There's an interesting one that I'll actually ask either of you to maybe jump in and take this one. One of the questioners, and I'm not sure what the source of this information is, but it says that COVID-19 severe patients have high levels of IL-10, which is known to be an anti-inflammatory cytokine. How do you reconcile that, or can you reconcile that with your IL-10 data? I think that was from your presentation, Raquel. Any thoughts on that, or is it relevant?
Yeah, IL-10 is definitely some cytokines. I can say they are dual and they have both pro-inflammatory and anti-inflammatory properties. I'm not familiar with this exact data that you're presenting. The only speculation that I can make here, that maybe in some of the patients, there is an elevation of IL-10 in response to the inflammation and in attempt to, the body also tries to fight the inflammation. So maybe in some of the patients at the time point that they were checked, there was an attempt of the body to fight this inflammation, but this is a pure speculation. Eric, if you have other thoughts?
Yeah, no, I think that's probably right. I mean, I would echo that. There's a lot we don't know yet about the human immune response to these viral infections. But I think it's quite clear that there are both redundant mechanisms that support the hyperinflammatory response. There's also counter-regulatory responses to try to control the inflammatory response to an infectious disease to prevent it from getting out of control. And clearly it's the disruption of these regulatory mechanisms that lead to conditions like cytokine storm and ARDS. But that's not to say that those counter-regulatory mechanisms are completely absent. They were just apparently insufficient in a subset of individuals to prevent those conditions. So I don't think it's really surprising that we see elevations of anti-inflammatory cytokines alongside elevations of inflammatory cytokines in these patients.
Okay, yeah, very great points. And thank you for taking that question. I know that you necessarily had to speculate there. So speaking of speculation, we have time for one final question for both of you. And I'll understand if you don't want to answer this one, but a lot of people are excited about this, and we had, of course, questions about timeline. And are you able to say when you anticipate beginning enrollment in the trials? And the second part's even more difficult, I guess, in the current circumstances to know, but do you have an idea about how long these trials might take?
I mean, the first one, we've already begun enrollment in our clinical trial. So we have enrolled patients into the trial, and that is progressing well. We've had a lot of interest from sites who wish to participate. So we anticipate that onboarding of sites can move quite expeditiously. But, in terms of completing trial enrollment, it all depends on the progress of the outbreak to a certain extent. And, we all hope the outbreak will go away and we'll never be able to enroll the trial. But I unfortunately don't think that that's the case. And we probably will see it enroll fairly quickly. I can't really provide, more concrete timelines than that. And I think, folks probably understand why, just because of the uncertainties of not only, clinical operations on a normal day, but in the face of an emerging disease outbreak with an unknown and unforeseeable progress. I think that would be imprudently speculative.
Understood.
Yeah, so we are about to begin. We have open sites and we are in very late stages of setting clinical trial. I don't want to provide a date, but it's there in the US. And regarding the timelines, my turn to echo Eric. We hope to move very fast. We think we have a chance to recruit very fast, but I cannot provide timelines.
Okay. Well, thank you for those answers. I think, and unfortunately, I don't think you're going to have any trouble enrolling patients for this. So we look forward to hopefully seeing some positive clinical results. So with that, we're a little bit over time, so I'm going to close it out now. I wanted to thank both of you for fielding all those questions. I want to thank the audience